Today, I'm going to talk about protein structure. Those advanced learners shall not listen to this because this is for very beginning level learner. So, I repeat, those who are starting to study for CSIR, those who are in their master's courses, or those who are just to start masters can listen to this. This is very elementary information, but this is very essential because without this knowledge, you cannot study further. So, this is the foundation. So, in the last class, we learned about amino acids and the structure of amino acids. We learned how to memorize the names and how to memorize the structure of amino acids, the 20 standard amino acids. So in this class, we are going to learn about proteins. So proteins are made up of amino acids, or amino acids are the monomers of proteins. Proteins form a very important class of biomolecules due to their role in the structure and function of a cell. Aquaporins, which are important in entry of water into the cell. Beta adrenergic receptor involved in the cell signaling process. DNA polymerases and RNA polymerases involved in the synthesis of DNA and RNA. Hexokinase present in the uh, glycolysis. Rubisco in photosynthesis. Collagen protein, ATP synthase, albumin, and the number is infinite. I mean, the cell is made up of proteins as a major component, and many of the enzymes are proteins. So, without protein, the biological functioning of a cell will be very difficult. All these proteins are made of just 20 amino acids. Why? Have you ever thought why the 20 amino acids make the entire plethora of proteins? Why? There are other amino acids also, right? So, if you look into the uh, synthesis of protein, you can see that the DNA molecule makes RNA, the mRNA molecule, by the process transcription, and the mRNA will be processed, then later ribosomes are attached to it in the cytoplasm to synthesize proteins by a process called translation. And these proteins are later modified to functional proteins. The amino acids are connected to each other by means of peptide bonds. So the covalent bond that connects amino acids together to form protein is called the peptide bond. So this is how it is formed. You can see the one amino acid on the left and the second amino acid on the right upper side. And you can see in the yellow color that two molecules of hydrogen and one molecule of oxygen. So it condenses to form water. Okay, so one more molecule of water is eliminated from it, and as a result, a new bond. Here in the second diagram, you can see the green color. So that new bond is formed by eliminating one molecule of water. The water molecule is shown in the blue bubble in the downside of the diagram. So this bonding that occurs between the carboxyl end of one amino acid and the amino end of the second amino acid is the peptide bond. It is planar in nature. So this is the process. So you have one amino acid which will be connected to the second amino acid by peptide bond. Third amino acid will be joined to it, followed by the fourth, fifth, sixth, seventh, eighth, ninth, 10th, and it goes on. So this is the way the amino acids are linked to each other to form a protein. So this basic connectivity between amino acid is called as the primary structure of amino acid, right? So which bond stabilizes the primary structure? Of course, the peptide bond. So secondary structures, when you see the primary structure of proteins, they don't sit simply. They form further complicated structures. They get coiled. They form helixes. Okay, helical coils called as alpha helices. Okay, alpha helices. So these alpha helices are connected to each other by means of hydrogen bonds. Okay, so this is another secondary structure. You can see here, this is not a coil, this is a sheet-like structure. You can see one layer of protein is connected to another layer of protein by means of again hydrogen bonds. This is called a beta pleated sheet, right? So which bond stabilizes the secondary structure of proteins? Yeah, hydrogen bond. So this is the next level of organization of proteins. So first we saw primary structure, then secondary structure. Now this is the tertiary structure. Okay, so what is this? So in this, you can see that alpha helices, you can see, which are forming further coiled and folded structures. Okay, so the further complex organization of secondary structures into folded structure is called tertiary structure. Okay, so this mainly occurs due to the presence of hydrophobic amino acids. The hydrophobic amino acids form the central core of the tertiary structure. Meanwhile, hydrophilic amino acids are arranged in the peripheral zone of this protein. So, and another one is disulfide linkages. So you can see the disulfide linkages also connect between two cysteine residues in an amino acid. So this is the quaternary structure, the fourth level of organization of protein. So as an example, I can say hemoglobin. Hemoglobin is made up of four globin molecules and one heme. So heme acts as a central point, right, to connect these globin molecules. So the blue and red and all are uh, separate units, separate tertiary units, which are united to form the quaternary structure. So this is a summarizing of how these structures are formed. In the first diagram, it is the primary structure. When the primary structure gets coiled or pleated, it forms a secondary structure, and then it gets further folded to form the tertiary structure, as you can see in the third diagram. And several such tertiary structures unite to form the quaternary structure. So this is all I wanted to tell in the as a very introductory lecture to the organization of proteins. So further advanced informations for the CSIR examination will be given in the next part of this lecture. So YouTube gives you notification if the channel is subscribed.